talking about DNA, DNA structure, DNA replication, and now it is time for us to talk about DNA and protein synthesis and how these two come together. So how do we get proteins synthesized from DNA? So let's go over the steps that we have here, and then we'll go into a little bit more detail in some of the other slides. So first off, we know that DNA must be transcribed into mRNA or messenger RNA. So in order to have a protein to be made, what we have to do is first start with creating an mRNA strand from DNA. Now this mRNA strand will leave the nucleus, it will travel to the cytoplasm, and it would then go to those ribosomes and that are in the cytoplasm to use the code that's on that mRNA to translate it into an amino acid, which if you remember, amino acids are those the basic building blocks of proteins. So once you have a whole bunch of amino acids put together, then you're going to have a protein. So over here on the right, I also have a little bit about which step is which. So we're first going to talk about transcription, and then we're going to talk about translation. So if you want an overall view, make sure you come back to the beginning of this video. But if you want individual steps, we're going to go talk about those right now. So the first step is something called transcription. So if we want to build a protein, we first have to transcribe DNA into mRNA or messenger RNA. And the reason for this is because DNA is too large to leave the nucleus. So DNA can't just make a protein. We have to have kind of an in-between little step. So if you remember in one of our previous videos, I mentioned the whole analogy about how DNA and proteins don't speak the same language. We sort of have to have a translator, and that's what mRNA does. So we first have to go through the process of transcription, or basically this DNA strand has to be transcribed into mRNA. Now, transcribed or making a transcription is really just making a copy of it. And in this case, we're taking DNA and we're making a copy of it in an RNA strand in order for it to leave the nucleus. So transcription occurs in the nucleus, and that's because that's where the DNA is. Now, just like when we were talking about DNA replication, helicase is going to be that enzyme that unwinds the DNA. So it's going to look more like a regular ladder rather than a double helix. So it's going to unwind the DNA. And it's going to break that the rung apart. So the rungs of the ladder get broken apart. Now, to create mRNA or this messenger RNA, what happens is we have RNA nucleotides pairing up with the DNA strands. This is similar to what we were doing in DNA replication, where we had a complementary base coming up to make a semi-conservative new copy of the DNA. Now, in this case, we have RNA nucleotides, which follow the same base pair rules for the most part, except for uracil is actually used in place of thymine. So uracil is actually going to pair with adenine. So if you take a look here below, if we were to take this DNA strand that you have here, and we want to transcribe it into an mRNA, we have to make sure that we have the complementary base pairs just for an RNA strand. So we still know that T will go with A, G will go with C, C will go with G, but here's the difference. If you notice, A is actually going to U, and that's because this is the complementary base pair in RNA. Now, once we go to the next one on our DNA strand, we have a T, but that still goes with A. So the only difference that we have here is T on our complementary base pairs are going to be exchanged out for U. And it will continue down the line. See, we have another uracil here. G with C, and then we have another uracil here. So when we're transcribing DNA into an mRNA strand, please be careful of that base pair rule where uracil will actually pair with adenine. Now, if we are doing it backwards, don't forget T still goes with A, but A will go with U. So just as a quick overview of transcription, we have DNA here. We have a DNA strand that is going to be unwound by helicase. And if you notice, we have the RNA nucleotides coming together here in order to make a copy, but this copy is actually messenger RNA, which is then allowed to leave the nucleus. So transcription will begin on the area of the gene or the area of the DNA that contains the gene. Now, it doesn't just start anywhere. Transcription will actually start with a promoter. So transcription will start with this promoter gene right here. And the promoter is basically just a starting point. It's going to turn the gene on or off depending upon what it wants to code for. So that promoter region says, hey, start here. The second region here is the coding region, and this is the part of the DNA strand that is being transcribed. So it's quite possible that not all of the DNA strand gets transcribed. It's just that some of it might. And then we also have a termination sequence. So obviously termination means end. So your termination sequence is going to signal the end of the gene that is going to be transcribed. So in this picture here, if you see just this part of the DNA strand for this particular protein or this particular string of proteins is going to be started. So we have somewhere on each side, it doesn't really tell in the picture, but let's say we have the promoter region is going to start right here. So my awful P for my promoter region, we have RNA polymerase, which is actually works very similarly like we saw DNA polymerase in DNA replication. DNA polymerase is going to place those bases back together using RNA instead of DNA. So it's going to put those complementary base pairs together. And this little arrow shows that it is moving in this direction. And so somewhere over here is going to be the terminator sequence, and that is going to signal that the end is here, and that's all that needs to be transcribed. So as kind of a summary, remember we have a promoter region. We have an area that's being coded. So in this particular area, this is where RNA polymerase is going to bring those complementary base pairs together according to the RNA base pair rules. And then we have a terminating sequence, and that is going to stop the transcribing of this particular DNA strand. So just for quick practice in your notes, go ahead and jot down the complementary RNA molecule for the following DNA sequence. So remember, base pair rules do matter here. So take a few seconds to do that. Now, what you probably should have gotten is our A's go with T's, C's go with G's, our C is a G, our A is going to be a U for uracil because we're transcribing it into an RNA. We have an A here. My awesome drawing, C is going to go with G. My A goes with a U, G with a C, my A with a U. So you should have something regarding or looking like this sequence right here. So we've talked about transcription up to this point, basically where that DNA strand is going to be transcribed into an mRNA strand. That mRNA strand is going to then leave the nucleus in order to go to the ribosomes to do the next step. So because DNA and proteins don't have that same language, the RNA or that mRNA strand is actually the translator. So we're going to take that RNA strand, which we have created, which in this picture is this bluish gray looking line right here. So this is our messenger RNA. it is going to come up to the ribosome. Now, these are the free ribosomes that we see inside the cell, not necessarily the ones that are on our rough endoplasmic reticulum. So we want these ribosomes to be free in the cytoplasm. Now, these ribosomes consist of two subunits of rRNA. So we're going to talk a little bit about other types of rRNA. So we have ribosomal RNA, and the ribosomal RNA is actually the stuff that's in the ribosome. So that's already there. In order to translate the message of that mRNA, we have to have the actual translator, which is something called tRNA or transfer RNA. So this guy right here in the picture, these little ones in yellow, those are our transfer RNAs or our tRNAs. Now, if you take a look here, tRNAs look kind of funny. They have a sequence down here on the bottom here. Notice that there's a 3-coded sequence on our tRNA. And also at the very top, we have these 3 letters that actually are the proteins, or I should say the amino acids. So these amino acids right here will eventually grow into the protein chain. So the tRNA acts as the translator, and these molecules, these tRNA molecules, bring the correct amino acid to the ribosome so the mRNA can read it. Now, you see on our mRNA, we have these little things here, and we have these things, and I'm circling them right now, and these are called codons. So on our mRNA strand, we have 3 letters that act as something called codons. Now, these codons are very important because they are going to match the 3-letter code that is on our tRNA right here, and those are called anticodons, and we're going to talk about those in the next slide. So keep this in mind as a summary of this particular process. We have translation occurring at the ribosomes in the cytoplasm. We have these ribosomes containing rRNA, but the big one that's really important here is tRNA, because tRNA acts as that translator. It's going to translate that message on the mRNA because it's going to bring the correct amino acid to that mRNA in order for a correct amino acid to be formed. And here it is again, just in a larger version for you. We have these codons, we have an anticodon. So come back to this picture if you just need a quick visual. So I mentioned those codons, and codons are extremely important because translation will begin and they'll end at a specific start codon and a stop codon. So just like we saw in the beginning where we have a promoter region, we have a termination sequence, codons have something very similar. So codons actually do have a start and a stop codon as well. And we're going to read some codon charts here momentarily. So a codon is a 3-letter code for an amino acid. This is on the mRNA strand. So your codon is on your mRNA. So for example, we have AGU, or perhaps we have CCA. So your codon is on the mRNA sequence. So if you follow this red line right here, the codon is on that mRNA sequence that's right here. So in this picture, our codon would be AUC. Now the anticodon is on the complementary pair of that tRNA. So the anticodon is on your tRNA, and I'm circling that for you here. And if you notice the red line that comes out, here we go. We have our anticodon right here. That should be a complementary pair to the codon. So notice that A is paired with the U. This U is paired with an A. This G, they're paired together. So let's take a look at how to read a codon chart. We're going to do more of this in class, but I just do want to kind of mention it here, just real quick. So to read a codon chart, to figure out what amino acid will be placed on that once we were to kind of go through the process of translation, we first want to read the first base in the codon. So if you take a look at this first one right here, let's take a look at UCU. So UCU, the first U right here, that's our first base in the codon. So I'm going to find U up here, and I'm going to know that somewhere in this big pile, that's where I'm going to find my amino acid. So let's take a look at the second codon. I'm going to go to the second base in the codon is a C. So I kind of know that I'm looking in this big spot, but it's kind of down to just this particular column. Now we can already take a look. They're all SER. So we probably know that that's going to be our amino acid, but just to be sure, always look at that third base. So this third base here says U. I'm going to look at the third base column, and here's my third base, which is right here. And so when I follow all of these together, I am going to come up with this amino acid right here, which is serine. So I'm going to, if I was to try to figure out what's being translated, this would be the amino acid that gets translated here. We'll be practicing more of these in class. So I just wanted to mention that that is how we read it in case you weren't in class to get this, but we'll be practicing more in class.